welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. Now, we will go to lecture number 32, where I will talk about engineered yeast brews precursors of anticancer drug vinblastine. If you remember in a few previous classes, when we talk about tropane alkaloids as well as isoquinoline alkaloids, there also I have shown you examples, rather very positive achievements using yeast system to synthesize the either benzyl isoquinoline or tropane alkaloids de novo. So, the similar attempt was also made with this benzenoid indole monoterpene indole alkaloids, leading to formation of the precursors of vinblastine. So, we will briefly look into this aspect today, that is de novo synthesis of dimeric monoterpene indole alkaloid precursors in yeast cells. Let us go to the board now. Well, so the topic what I am going to write, a microbial supply chain for the production of the anticancer drug vinblastine. So, this work what I am going to discuss now, this work was published in the famous journal Nature just last year, and reference is volume number 609, page number 341 to 347, and the authors are Zhang and Kiesling. So, they are mostly based at Danish Technical University of Denmark. Okay. So, what basically the genesis of this work? So, that we will discuss. First of all, that why there is a need for microbial supply chain for the production of anticancer alkaloids, that is important to know. So, that means that we will now outline the problem. Problem is this, that the precursors of vinblastine, that is caferanthine and vindoline, as well as the final alkaloid vincristine or vinblastine, they are always harnessed from the plant tissue. Therefore, in order to extract this, you need a huge supply of this plant material. Now, because of different reasons, including natural calamities, political changes in the political scenario, sometime this pandemic, okay, forest fire, all this leads to the disruption of this supply chain. And if there is a disruption of the supply chain, there will be indeed shortage of this life saving drugs. Like shortage of drug means the shortage of the raw material for the production of this drug. Therefore, it is important to ensure the stable supply chain for these value added products. Now, what is will be the solution then? Solution will be using synthetic biology approaches where if possible the whole pathway can be transferred into a eukaryotic system, preferably yeast, and so that the yeast will be able to produce the desired products. And also it is important that ideally one should be able to make this recombinant yeast synthesize the desired alkaloids de novo. That means, you should aim not to supply the expensive precursors, rather you should only grow the yeast with basic sugars, medium and amino acids, and yeast will utilize that. It will run its primary metabolism. From there it will start the specialized metabolism, and ultimately the pathway for this monoterpene indole alkaloids leading to formation of the products. So, that is what it is. So, in order to make that, so scientists they made their task jotted down, and in 3 modules, that is they should try to complete each modules. So, the module 1 is basically the de novo synthesis of strictocidin. Then module 2 will be de novo synthesis of catharanthine, and module 3 will be de novo synthesis of vinblastine. So, each of these modules must be expressed in yeast, and tested individually to confirm functional expression of that portion of the pathway, So, and from here the final strain should contain all the 3 modules leading to the formation of catharanthine and vinblastine using glucose and amino acid tryptophan. Well, so this should be. So, let us briefly draw the modules what needs to be operative in the yeast. So, the module 1, say the strictocidin module, if you say. So, I just first write the sorry. So, this makes loganic acid. Loganic acid will make loganin. Loganin will make secologanin, and this subsequently will make strictocidin, and then it will make strictocidin aglycone. This all you know because I have said this repeated times, but this is required to tell you once again. Okay. So, this is the first module, that means from the geraniol to the formation of strictocidin aglycone. So, this is the strictocidin module. Sorry, STRI. Well, so the next step, it makes geisocyzin, and then it eventually makes stemadenin. This stemadenin will be converted into stemadenin acetate, and this will be converted into precondylocarpine acetate. Now, this is important because the scientists really they were waiting until the two missing enzymes are discovered. So, these are PAS, precondylocarpine acetate synthase, and DPAS, dihydro precondylocarpine acetate, and this will leads to the formation of tabarsonin. So, and stemadenin acetate also contributes towards the formation of catharanthine. So, the module 2 is basically from here, that is strictosidine aglycone onwards until the formation of catharanthine and tabarsonine. Okay. Now, this is the module 2. So, what you should write here, that is tabarsonine catharanthine module. Now, the module 3, which is basically the vinblastine module. So, this will start from tabarsonine. This all you know, but still I would like to put it here, that is tabarsonine will make 16 hydroxy tabarsonine and then 16 methoxy tabarsonine and then subsequently it makes 3 hydroxy 16 methoxy 2,3 dihydro tabarsonine. These steps all we have studied, and this is by the action of these two enzymes, and this leading to the formation of D-acetoxy vinblastine and then D-acetyl vinblastine and finally it makes vinblastine. Okay. So, let us circle it or let us make the border for this, that is from anyway I am not telling anything new. So, already I have discussed this, but just to recapitulate your concept, I have drawn this once again. Okay. Now, 3 modules were individually expressed in the yeast, and finally, as I said that finally, the spinal strain they have constructed which contained all 3 modules sequentially, and that if function, that leads to the formation of catharanthine and vindoline. Okay. So, the question you should ask yourself, why they basically stop there, why not to add a peroxidase in the yeast system, so that both the catharanthine and vindoline produced in the yeast system can be utilized for the production of vincristine. That means, if we put line here, that means catharanthine and vindoline, they should join and then they make vinblastine, and this is peroxidase, sometimes people mention like PRX1. But in spite of several attempts, they fail to express peroxidase functionally in the final yeast strain. That means, the final yeast strains were incapable of producing vinblastine. Now, that is basically the bottleneck. That is why at this stage, using this recombinant strain, where all this altogether they made 56 genetic edits. So, let me go to the next slide. So, that means, in the final yeast strain, they made 56 genetic edits, including (34) genes from plants and deletion of or overexpression of 10 genes of yeast origin. So, this part we did not I did not show you in the previous board, because to make the things simplified. And why all they did? They did all this basically to improve the generation of key precursors in the biosynthetic pathway. Okay. So, the what will be the if we put it in the form of a diagram, say the wild type yeast this is wild type, and from this wild type is what was achieved at the first stage is basically the de novo strictocidin. This is okay. From there they made this is de novo Okay, I think I should give the space here. Tabarsonin. From there, the next de novo vinblastine. From there, de novo catharanthine and vinblastine. These are all yeasts. So, this is successful, but why they are unable, they fail to express peroxidase in yeast strain. So, what they thought, perhaps it requires more compartmentation, and what scientists did for isoquinoline alkaloids. So, basically they utilize the peroxisome and they put some enzymes like narcochloride synthase inside the peroxisome. So, now what they thought, that perhaps they will put the peroxidase enzyme inside the peroxisome and drive the pathway there, so that may be a safer subcellular compartment which will allow the peroxidase to function, and that eventually may lead to the formation of vinblastine. But in order to achieve that, we have to wait. Okay. So, that is basically the future direction. So, the future direction, I will end this with writing future direction. To have the peroxidase expression inside subcellular organelle, and that probably help to couple the catharanthine and vinblastine together and makes the vinblastine. So, whatever achieved until this, that is to make stable production of vinblastine and catharanthine. So, that they have achieved, that itself is a very great achievement. That is why the paper published in Nature last year. So, with this I am basically ending this class. So, the de novo formation of vinblastine precursor in yeast cell is now achieved. With this I end the class. Thank you.